Hello, welcome to this oral session of Organization for Human Brain Mapping. It's bright and early my time, but for some of you it's in the evening. But welcome to this session on neurodevelopmental disorders and environmental impact. Today we have three talks that cover autism spectrum disorders, one looking at harmonious family function, and also one on cannabis use and effects on the brain. So the first talk will be a multi-analysis approach to task-modulated functional connectivity in autism by Caroline Musnung. The second talk will be gray matter co-alteration networks in autism spectrum disorder, a meta-connectomic approach by Jordi Manuelo. The third talk will be aberrant social orienting and extrinsic functional connectivity during natural viewing in autism by Juha Lanakoski. The fourth is functional cartography of cognitive dysfunction in focal epilepsies, a multi-scale task fMRI analysis by Lorenzo Cacciagli. Then harmonious family climate mediates the impact of socioeconomic status on child brain function by Han Zhang. And then finally, cannabis use during adolescence is associated with altered cerebrocortical development by Matthew Albal. I'm very looking forward to these talks, and we welcome you again on behalf of myself and my co-facilitator, Michael Millam.
So thank you very much for the opportunity to present on this very special OHBM conference. So the data I present today are part of the Longitudinal European Autism Project. In this sample, or in this project actually, a sample of over 400 individuals with autism and 300 typically developing individuals aged between 6 and 30 years have been characterized extensively on multiple levels, including brain structure and function. And in this talk, I want to focus on one of the core deficit domains of autism, which is social communication deficits. So we included two functional tasks that tap into this domain. So in one task, subjects watch a cartoon depicting two triangles which move about the screen and elicit a spontaneous mentalizing. In the other task, subjects are asked to match emotional faces. And using this large data set, we followed up on the question whether autism is characterized by alterations in large-scale functional connectivity networks involving social brain regions. So we tackled this question actually using conventional mass univariate approaches and a novel multivariate analysis approach, which I will go into detail in a second. Using these methods, we were interested in the effects of task, sex, age, and diagnosis. We were also interested in the effect of daily adaptive functioning. So this adaptive functioning is one of the core autism measures, actually outcome measures in autism. And a recent study in our group actually have demonstrated that social communicative deficits are tightly linked to lower adaptive functioning. So in the conventional general linear model approach, we selected key regions of the social brain network and performed a brain-wide voxel-wise psychophysiological interaction analysis, the PPI analysis. This analysis highlights brain regions that show task-modulated coupling and decoupling with the seed region. The top row shows the PPI results for the dorsomedial prefrontal cortex with hotspots in the inferior frontal and angular gyrus, both of which have been associated with the mirror neuron network, for instance. And on the right side in the top row, you can see a scatter plot. This scatter plot shows the strength of coupling for the peak voxel in the angular gyrus, and you can see very clearly that this is not associated with age. And The violin plots on that side also show that the effects of coupling or decoupling are not associated with diagnosis or sex. So these scatter plots and violin plots have been extracted from this angular gyrus voxel, but I can tell you, not showing here, that this is the fact for the full brain. And on the lower level of this slide, you can see the whole brain maps for coupling and decoupling for other regions as well, which we have used as seed regions, all of them being clearly classical regions of the social brain, such as the superior temporal sulcus or the fusiform gyrus. All of those, again, show clear task modulation, but they have not shown any effects of age, sex, or diagnosis. So results are quite similar for the emotion processing task. Seed regions here were located all in the inferior temporal lobe or cortex. So for instance, in the right amygdala or in the fusiform gyrus on the left or right hemisphere, again, we did not see any effect of age, sex, or diagnosis on the functional connectivity. But which is striking actually in these slides is that you can see a very pronounced decoupling of the seed regions and not a strong coupling or synchronization during the experimental phase of the task. This is certainly of interest, and we are following up on that in current analysis. So our second approach was to use a multivariate approach to study functional connectivity. The idea of this approach is that changes in functional connectivity are reflected by variations in covariance matrices. So the idea here is that you extract time series from parcels of the brain, and then you calculate covariance matrices. And the idea is now to look at fluctuations here. The SPADE is a short form for spatial patterns for discriminative estimation. And the idea is to estimate linear spatial filters. And these spatial filters are meant to discriminate as much as possible between two labels, for instance, task 1 versus task 2. And the evaluation or the resulting filters, the performance of those filters is evaluated first using a leave one out cross-validation to test which filter actually is informative. And then we are using a training and a test sample to evaluate or test the generalization of the performance of the filter using linear discriminant analysis. So the approach in the SPADE analysis is that we are looking at changes in covariance structure on a multivariate way, taking into account changes in covariance at all levels of the brain. Whereas in the GLM approach, which is the seeded connectivity we have used here, we are looking at the changes in mean signal. Very different, because here in the GLM, we are only focusing on one single voxel at a time. Now looking at the performance of the SPADE filters, you can very clearly see that SPADE was nicely able to discriminate between the two tasks. And you can also see that the areas associated with this maximal discrimination between the tasks, which we can translate maybe into unique fingerprints. They are associated with areas which we conventionally associate with emotion processing, processing of emotional phases or social cognition. Now looking at the discrimination ability of the filters for the between subject comparisons, you can see here that accuracy drops to the level of 70% for both tasks, for the age effects and for the sex effects. And it's dropping down to chance level for the discrimination between diagnostic groups. So this is very informative, because both very different methods suggest that the active brain during social information processing is not different between individuals with ASD and typically developing subjects. Now in my last slide, I want to show the association of discriminative filters with adaptive functioning, the outcome measure, which we were looking at. You can see here the distribution of the Vineland scores. The Vineland is the questionnaire we are using here to define social functioning or adaptive functioning. And the lower edge, this is the lower 25%, are individuals with low adaptive functioning, and here the individuals with high adaptive functioning. And the question is now, can we see a spatial filter or a pair of spatial filters which discriminates well between those two cohorts, so these two samples, high versus low scoring individuals. And indeed, only for this theory of mind task, the social cognition task, we can see a quite nice discrimination between those two sets of individuals. This is not the case for the face processing task. And when looking at those filters, we again can see that we have frontal, we have parietal areas which are contributing to this, but also, for instance, and that quite strongly, the dorsomedial prefrontal cortex, which is one of the key regions in social processing, in mentalizing, and which has previously been shown to be associated with autism traits. And with this, I want to close this presentation, and thank you very much for your attention.
Hello. I'm really pleased to have the opportunity to share and discuss with you the results of our work aimed to analyze autism spectrum disorders applying a pathoconectomic approach. So here is a brief outline of the talk. And so let's start briefly discussing why we think that this kind of approach could be useful in the frame of autism spectrum disorders. ASD surely is a challenging topic for neuroscience, considering the heterogeneity of the phenotypes inside the spectrum, the still unclear etiology, and of course the prevalence. Both structural and functional alteration had been previously reported and described in literature, and voxel-based morphometry studies and coordinate-based meta-analysis had highlighted patterns of gray matter and white matter alteration spanning across neocortex, the basal ganglia, and the cerebellum as well. However, we think that possible network-like properties behind these networks are still not fully appreciated and understood. So what does it mean to take a pathoconectomic approach? Well, it is not only to observe what happens to the healthy functional and structural connectome when a disorder bursts in two, but it is trying to understand if these patterns had some network-like properties behind. So it is to infer the existence of statistical relationships between the brain regions that compose these patterns. And this is why we propose to call these co-alteration networks. And in fact, one of the most interesting evidences from the field of pathoconectomics so far is that this kind of structural alteration does not spread randomly across the brain, but it is somehow constrained by functional, structural, and even genetic connectivity. And this is something that was recently confirmed by our team, even at a meta-analytic level and on a transdiagnostic sample. So how can we build this kind of co-alteration network? Well, this is a co-alteration network. This was based on Alzheimer's disease from a previous study of our team. And as in any network, we use nodes and edges to represent the information. In this case, the nodes represent brain regions that are structurally altered in the disease, while the edges express the frequency of this co-alteration between the two nodes that are joined by that edge. Since this is an approach based on meta-analytic level, the first step is to create the data set, and this can be done as another coordinate-based meta-analysis. In this case, we started querying the OHBM set of the brain map databases, and then we integrated the querying through PubMed. And at the end, we obtained a set of stochastic coordinates with the peak of effect. And so at the end of the screening, we got 80 OHBM experiments for both decrease and increase condition in gray matter. We decided to keep both the condition because both of them were widely described in literature. And so we included nearly 5,000 subjects in the analysis. Then the set passed through the anatomical likelihood estimation algorithm in order to estimate the spatial coherence between the experiment in the dataset. And from the resulting map, we built the set of nodes. And this was done using a peak detection routine and then applied a couple of extra criteria just to avoid the overlap between the nodes. And finally, we built the edges using the Patel's K index. And the K index measures the probability of co-alteration of two nodes against the probability of independent alteration of the two nodes. So this basically means that it tests each possible couple of nodes inside the network, and then it goes across the experiments. And for each experiment, it checks if these two nodes are both altered or only one of the two is altered or none of the two is altered. And then at the end of the whole samples, it draws the statistics. So how does it look like in the case of ASD finally? Well, we obtained two networks made of 28 nodes each. Of course, the nodes were different. And for the decrease condition, we had the same amount of interhemispheric and intrahemispheric edges, while for the increase condition, we have slightly more interhemispheric edges, but we can see that in both the condition, nodes and edges go across both the hemispheres. And we can also notice that we have lots of long-range edges. And so this is in line with the hypothesis of long-range impairment of the connectivity in ASD. And we can also see at first glance that the cerebellum is only involved in the decrease condition. Then, since the two networks were quite dense, we decided to apply the K-core decomposition in order to detect a subset of the nodes that is highly interconnected. Because the interesting point of this kind of analysis is not only to spot which are the regions that are structurally altered, because this is a kind of information that could be retrieved by using a standard coordinate-based meta-analysis. But here the relevant point is to understand the relationships between the nodes. And so in this way, we were able to appreciate the edges linking frontal regions with posterior regions. And this is again in line with the hypothesis of fronto-posterior underconnectivity in ASD. And on the increased side, we can see the role of the insula as a hub, even at the co-alteration level. And this is something that mirrors the functional connectivity already described in literature. And so this could suggest, again, a role of the insula in the switching between networks, and so an alteration of multi-system integration in ASD. And finally, we wanted to give a functional characterization to the nodes. So we allocated them based on known functional networks. And we can see that both increase and decrease condition converge in the default mode networks. And this is somehow expected based on existing literatures. But there are some networks that are mainly targeted in one of the two conditions. And this is sensory motor network for the increase, for example, or visual networks and limbic structures for the decrease. And this is coherent between the full network level and the K-core decomposition as well. So of course, we have to account for some limitation in this kind of approach. First and main one is that it was not possible to distinguish between potential different phenotypes inside the sample, and we avoided to split too much the sample in order to avoid to lose statistical power. And the other thing is that, unfortunately, at this moment, the co-alteration network that we can build are still undirected. So this means that we can detect two nodes that are altered together, but we can say if one is altered first and then the alteration goes to the other node, or if it is the other way around. However, to conclude, we can see that, yes, we identified network-like properties behind both the patterns, and we spotted the existence of cerebrocerebellar pathways even at the level of co-alteration. That is something that is well described for functional level in ASD. And then it's interesting also to note the existence of alteration with visual region, and this is something that is not often found for the co-alteration network in other disorders. So this could be something that is quite specific for ASD. And then, as we see, this kind of approach can help to add a new layer, a new level to understand autism spectrum disorder, and of course, any other disease that can be studied in this way. So before to conclude, I really want to thank the whole Focus Lab of the University of Turin that joined with me in this project, and Professor Roberto Keller that joined us for this specific work. And of course, I really want to thank all of you for your attention.
I'm Johan Harkistian. I'll be talking today about social orienting and extrinsic functional connectivity during natural viewing in autism spectrum disorder. Autism spectrum disorder is a developmental disorder that is characterized by impairments in reciprocal social interactions and repetitive behaviors and restricted interests. However, a severity of symptoms and learned abilities to compensate for social difficulties can actually vary quite widely within the same diagnostic label. This may actually also be reflected as developmental differences in underlying brain mechanisms, as is suggested by increased idiosyncrasy of brain activity during natural viewing conditions. However, diagnostic behavioral scales and most prior fMRI designs may not accurately reflect the individual social difficulties and differences thereof. So therefore, the goal of the current study is to elucidate the links between neural abnormalities and difficulties in social functioning. And to that goal, we started by observer-independent characterization of social behavior during dyadic interaction rather than relying on subjective evaluations using predefined scales. Then during fMRI, we analyzed inter-individual or extrinsic patterns of brain activity in response to viewing of socially relevant content presented in short movies. And then the big question or proof of concept in this study is, can we link the objectively measured behavioral abnormalities during interaction to actual brain activity patterns during social observation? So how we did this was, first, during diagnostic interviews, we measured with Kinect motion sensors the movement patterns of bodies and facial orientations of people, patients and controls while they underwent this diagnostic interview. And then we calculated the angles of the face orientation in relation to the interaction partner, both for the interviewer and the participant or patient. And to summarize this data, we collapsed it over time and built these two-dimensional histograms. So on the x-axis, we see the face angle of the interviewer, and y-axis, the angle of the participant. And the brighter the colors are, the more they spend jointly orienting in that particular orientation. So for example, if both had low angles at the same time, that's usually associated with eye contact behavior. And when both have larger angles at the same time, in this particular context, they are often jointly attending target on the table. During the fMRI, we showed a set of pre-validated movie clips depicting 5 different categories of content, so emotional scenes, neutral scenes, social interaction scenes, and non-interaction scenes, as well as painful scenes. And at the bottom, you can see the time course of the experiment. To analyze the functional connect or fMRI data, we used extrinsic time-varying phase synchrony. So essentially, we took the time courses from two different regions from two different participants and calculated the phase coherence of these two signals. And then when we repeat this for all pairs of regions and all pairs of subjects, we get this time-varying connectivity matrix that reflects how similarly at each time point these two or pairs of regions are activating. Then to the actual results, we see from the orienting results that orienting toward communication partner is reduced in autism spectrum disorder, and it also correlates with ADOS score. So the main effects you can see for the two groups on the left, and then their contrast in the second from the right plot, and you see that the low angles are higher in the controls versus patients, but actually, if we look at the correlations or partial correlations controlling for group differences, you see that in the right plot that the patients with very high actual autistic symptoms tend to spend actually more time looking at the interviewer. So this may be reflecting a overcompensation behavior that is often patients are taught that they should look at their communication partner, and some of them actually do it too much compared to you and me. To look at how similar these patients are and the controls are to each other, we created these correlation matrices of these orienting histograms, and then collapse this data over the columns to get a single value of behavioral typicality there on the right of each person. What we see here is there is quite high variability between the patients marked in red compared to the controls, who are very similar to each other, both in their behavioral typicality and their ADOS total scores. In the fMRI, looking at the big picture, we see that both groups show extensive networks of visual and higher order regions that synchronize. However, there is anti-synchrony originating from the cingulate cortex in the neurotypical controls, as shown by the blue dots in the limbic to occipital region. If we then look at the actual contrast between the two groups, now just looking at the significant links, we really see clearly these two effects. So there is negative effects and positive effects, which reflect aberrant functional segregation and integration of extrinsic processing in ASD. So these blue links are actually negative synchrony in the control group, which is absent completely or even positive in the patient group, whereas the red links show links that are generally positive, but they are just higher in the controls, so more idiosyncratic in the patients. If we then look at the time behavior of these links, it even more highlights this segregation or lack of segregation between these different connections. So at the bottom, you see the positive connections. The time courses are quite similar between both groups, but they are higher in the blue control group, whereas in the negative connections, the time courses are quite distinct. And in fact, in the patient group, the time course is very correlated, as shown by the correlation matrix on the right, with the positive connection. So they are actually forming almost a single network rather than being segregated as in controls. Now to look at the proof of concept results, we see that if we only look at these negative connections, there are significant group differences, as is defined by the analysis actually. because we are looking at links that do have group differences. However, if we regress out the mean group effects in the partial correlation on the right, there is no actual individual differences that would be explaining the connectivity in the ADOS score. However, if we then look at the behavioral typicality, we also see this correlation of group difference on the left scatter plot. But when we then control for the group difference, that actually doesn't change the correlation so much. So it remains negative, though not significant with this particular group size. So in conclusion, we see that in ASD versus neurotypical controls, the patients orient less toward their interaction partner. However, patients with high ADOS scores tend to orient actually more toward the partner than those with lower scores, potentially reflecting overcompensatory oriented behavior. Overall, global stimulus-driven functional network patterns are largely consistent in both groups. However, there is aberrant segregation of posterior cingulate and precuneus default mode regions from sensory regions in the patient population. Overall, we see also that ADOS total scores do not predict individual differences of connectivity controlling for group only. The group difference of ADOS predicts the connectivity. However, it seems that behavioral measures show promise for individual prediction beyond group differences. So we just need further study what is the best measure to catch these differences. So future studies will verify the reliability and specificity of these results and also compare against the other social interaction disorders. So thank you for listening, and of course, thank you to all the collaborators who made this work possible.
So thank you for the opportunity to present my research here. My name is Lorenzo Cacciali, and I'm a clinician scientist undertaking postdoctoral training at the University of Pennsylvania, Penn, supervised by Dr. Danny Bassett. I will be presenting results of collaborative research among UCL, Penn, and the MNI, focusing on the neurocorrelates of cognitive dysfunction in focal epilepsy. So epilepsy is a common disabling neurological disorder and consists in an enduring propensity for epileptic seizures, along with the accompanying cognitive and psychosocial consequences. Epilepsy is a very heterogeneous family of disorders, and in this talk, I will focus on the two most common focal epilepsies, i.e. epilepsy types in which a specific hotspot for seizure onset can be identified. First, temporal lobe epilepsy, TLE, the most frequent epilepsy syndrome overall, with seizures arising from the hippocampus. And second, frontal lobe epilepsy, FLE, slightly less frequent, but quite problematic clinically, with seizures arising from frontal lobe locations. Cognitive dysfunction is a major comorbidity of epilepsy. A long-held view here posits that the core pathological change, i.e. the presence of a temporal or frontal lobe seizure focus, determines the associated syndrome-specific cognitive profile, so more memory impairment in TLE and more executive dysfunction in FLE. More recent evidence, however, indicates that the matter is slightly more complicated, with widespread patterns of dysfunction seen in both syndromes that may be largely shared. As for the neurocorrelates of these cognitive profiles, while several fMRI investigations documented frontotemporal alterations during language or memory in TLE, our understanding of cognitive network organization in FLE is very limited. In this study, we thus aim to bridge this gap of knowledge, and first, we set out to investigate cognitive function and task-related functional repertoires in FLE compared to controls, using an extensive neuropsychological test battery, as well as 2 language and 2 working memory fMRI tasks. For language, we used covert verbal fluency and verb generation tasks that both elicit robust frontal lobe activation and, particularly for the verb generation task, also lateral temporal activation, owing to its semantic component. For working memory, we employed one verbal and one visual adaptation of the N-back task, the latter entailing 2 levels of difficulty, 1 and 2 back. Both tasks elicited the typical frontoparietal activation, along with deactivation of the DMN. To then extend our investigation and establish a functional cartography of cognitive function across focal epilepsies, we aim to directly compare and disentangle differences between FLE and the better characterized CLE to answer the question of whether an epileptic focus in the frontal versus the temporal lobe may result in similar versus different cognitive network profiles. In addition to controls, we recruited 56 patients with FLE and 64 patients with TLE, among the largest cognitive fMRI samples in epilepsy reported yet. Patient groups were comparable for clinical variables, and we controlled for age and sex in all group comparisons. FLE and TLE had weaker performance than controls for all cognitive measures, tapping into verbal and language processing, working memory, and executive function. Differences between TLE and FLE were found for naming, weaker in TLE, and for mental flexibility, worse in FLE. Working memory task performance was equally impaired in both groups, particularly for the 2 back conditions. For our multi-scale functional mapping approach, we started by analyzing voxel-based task activation maps, general linear models, and then summarized task activity patterns in the form of beta weights across 7 canonical functional networks, the 7 neocrinian systems. Third, we profiled task-related changes onto the principal functional gradient from Margulis et al. That describes a continuous transition from unimodal sensory to high-order transmodal areas, including DMN nodes at the opposite end, and reflects a hierarchical principle of whole brain connectivity. For our analysis, the gradient was divided into 20 consecutive bins. Task-related beta maps were aligned and basically overlapped onto the gradient, followed by a quantification of task-related signal per gradient bin. Altogether, these 3 scales offer a local to global transition framework through which one can interpret task-evoked changes and group differences. So, coming to our results, as a general remark, the upper rows show findings in controls, while group comparisons are more prominently shown below. For verbal working memory, we found reduced frontoparietal activation and failure to deactivate DMN areas in FLE compared to controls. Direct comparison of FLE and TLE showed more prominent dysfunction in TLE. So here, the warm color for patient group differences means higher activation in FLE than TLE. Blue would mean lower in FLE. Analysis across EO networks highlighted deviations of mostly dorsal attention and frontoparietal network activity from control data in FLE, here depicted in red, and even greater contractions in TLE, here depicted in orange. The black heptagon corresponds to reference values and controls. There were significant differences between TLE and FLE for dorsal attention activity. Profiling changes along the gradient revealed a global deviation of task-related recruitment from controls in both FLE and TLE, or in red and orange respectively, mostly for the middle gradient portion corresponding to perceptually guided and transmodal cognitive systems. Here, the area between the curves of FLE and TLE was also larger than expected from a null distribution. For visual working memory, there was slightly increased frontoparietal activity for low-level task demands in FLE versus controls, whereas for higher working memory loads, accompanied by performance drops in patient groups, we found marked reductions of frontoparietal activation in FLE, which likely mirrors defective recruitment of additional neural resources. Findings in TLE were fairly similar, and in fact, as shown here, there were no major differences between FLE and TLE. These patterns were also evident on a network scale, with hyper-recruitment for lower and contraction for higher cognitive load, as well as on gradient profiles, higher here for the easy condition in where TLE and FLE were very similar, and lower here for the high demand condition, particularly in FLE, with differences between patient groups for the area between the curves. As for language, verbal fluency first, FLE showed reduced activation of frontal as well as temporal language relevant areas, along with failure to deactivate DMN regions, mostly in midline. Activation differences between patient groups were limited. Analysis on canonical networks confirmed widespread activity reductions over frontoparietal, salience, and limbic systems, and global deviations from the norm for activity profiles on the gradient, particularly again in its middle portion. Here, while both patient groups deviated from controls, changes in FLE substantially mirror those in TLE. Finally, as for the verb generation task that has an expressive and a semantic component, FLE showed more localized activity reductions in frontal language areas, along with failure to deactivate DMN nodes, particularly in its lateral aspect. Here, on the other hand, profiles were less favorable in TLE, with reduced occipitotemporal and parietal activity compared to FLE. Deviations were particularly evident on canonical networks, with far greater activity contractions in TLE, and significant differences between FLE and TLE were evident basically across all task-positive networks. Deviations of activity profiles on the gradient were evident for both patient groups compared to controls, but slightly more so in TLE, with this area between the curves, between FLE and TLE curves, differing from a null distribution. So to summarize all these findings, our complementary analysis that track the neural correlates of cognitive dysfunction from a local to a network-based to a whole brain scale indicate global disorganization of cognitive networks in FLE as well as TLE compared to controls that affects both perceptually guided, so attentional, and transmodal cognitive networks. As seen for working memory, cognitive network saturation may occur prematurely for low-level task demands. And we also see failure to deactivate DMN areas across tasks and syndromes, which may point to a disrupted allocation of cognitive resources between task-positive and task-negative networks, and potentially represents a shared marker of vulnerability across focal epilepsies. Though patterns of dysfunction largely overlapped across syndromes, TLE appears accompanied by more prominent network disruptions, particularly for tasks involving verbal semantic processing, as evidence for the verbal working memory and verb generation language tasks. On balance, differences between syndromes are less marked than commonalities, and so on a spectrum ranging from no differences to maximum differences, I'll probably place my pointer around here. This study, of course, was a team effort, and I would like to thank my incredible mentors, Boris, Danny, and Matthias, for their support on this project, my colleagues and collaborators for their contributions, and quite a few funders, including the NIH, for supporting my postdoc. Thank you all for listening, and I look forward to your questions.
So hello, everyone. I'm Han Zhang from the Department of Biomedical Engineering of National University of Singapore. It is a great honor to share with you my work regarding socioeconomic status, family climate, and child brain function. First of all, when talking about SES, or we try to study SES, the most concern derives from the socioeconomic inequality, particularly income inequality. Based on the latest data released by the Organization for Economic Cooperation and Development, income inequality exists in almost every country within the organization. So why we should care so much about income inequality? Because low SES or low income could result in many adverse effects in our life and our offspring's development. For children, low SES could cause the delayed development of child cognition, such as language processing, episodic memory, working memory, etc. The delayed cognition or low cognitive ability of children could directly affect their educational achievement. For example, the World Bank data could tell that in a country with more population of low income, the nation's educational attainment is also lower. Besides cognition, low SES could also cause abnormal development of the child brain. Most studies until now consider the child brain structure. A typical example is that SES could affect the development of brain volume. Since low SES has so dramatic adverse effects on child development, the question now is, what could we do? Social science studies, particularly in the developing world, have suggested the promise of direct income supplementation. A lot of countries is trying so to use the national policy to directly increase the income of low SES families. However, in the future 10 years, the predictive modeling told us that there will be still hundreds of millions of people suffering from poverty. Not to mention the pandemic this year, the pandemic caused a worldwide economic downturn. A lot of people lost or are losing their jobs, and this effect could last for a long time. But the growth of children continues, no matter economic status good or bad. Besides supplementing family income, what could we try? Some papers suggested that good home environments providing cognitive stimulation enable children to overcome the effects of major adverse life experiences on cognitive development. For example, in low-income families, if the parent could emotionally support their children when children encountered problems, the children tend to have better cognition. Then, whether a positive family relationship could benefit the developing child brain. Here, the aim of this small work is to examine the potential protective benefits of good family interaction on child brain functional networks. We employed the baseline data of ABCD. We used the annual family income to stand for the level of SES. The child-reported family conflict skills was employed to evaluate the harmony of a family. In this skill, children were asked 9 questions, such as whether they feel that family members sometimes get so angry and they throw things. We also considered the child's general cognition by using the latent PCA scores generated by another study from our team with the ABCD data. The child brain functional networks were constructed with the resting state fMRI data. We firstly checked the association between SES, family harmony, and child cognition. It is clear that higher family income, better cognition in children. Similarly, a more harmless family, better cognition in children as well. The positive trend of family harmony and cognition stays the same with children from low, middle, and high-income families. By adjusted covariates, we applied the mediation analysis, which took the family harmony as the mediator. And we found the family harmony could partially mediate the relationship between family income and child general cognition. Then we examined the associations between SES, family harmony, and child brain function. We applied a functional atlas to parcellate the whole brain into almost 300 parcels, then constructed the functional connectivity matrix. The parcellation we employed could also be clustered into 8 causal level functional networks. Notably, different imaging protocols here were adjusted with an empirical-based approach. We found more than 800 connections that was significantly associated with family income. Most of these connections were within subcortical cerebellum regions, between subcortical cerebellum regions and motor network, as well as frontoparietal network. For family harmony, about 300 connections were found in relation to it. And the pattern is similar to what we observed in family income. Now the question is whether family harmony could be a mediator. To examine this, we specifically looked into 24 functional connectivities which were in relation to both SES and family harmony. We then applied the mediation analysis and found that family harmony could partially mediate the association between family income and 7 functional connectivities, which included the connectivities between the motor network and subcortical cerebellum regions, such as Piltman, as well as connectivities within cerebellum regions. And one connectivity is in the frontoparietal network. So to summarize, the results we obtained so far show that family harmony could partially mediate the association of SES with child cognition and child brain functional networks. So besides earning more money, holding a harmonious family is also very important for child development. Particularly for children in low-income families, maybe good family interactions could provide them a chance to lower the risks of their cognitive and brain development. So this is the small story I want to share with you all. With this great opportunity, I would like to express my gratitude to my respected supervisor, Prof. An Qi-Chu, for her guidance, patience, and support. And I also like to say thank you to my colleagues, Mr. Guodong Liu, who helps with data processing, and other lab members who provide good suggestions and technical support. And I really appreciate the ABCD data team to provide such valuable data. Here is our lab website and my personal website. All suggestions and discussions are welcome. Let's dive into child brain development studies and stay safe in this particular season. Thank you very much.